Hello everyone. So, as Professor Powell said, my name is Ilyas, and I work at the Institute of Physical Chemistry. I have a background in physics. I'm not sure I would call myself a physicist yet, but I work on trying to understanding how colonies of bacteria form, starting from single cells. So they divide and so on. Our objective is to couple modeling of these colonies with microscopy, and for that we use artificial intelligence. I mean, machine learning. Yeah. So, what about you, Vineeta?
Hi everyone. So this is Vineeta. I'm from India and I'm working as professor at the Institute of Physical Chemistry. And by profession, I'm a structural biologist. It sounds like a scary movie title, but it is not. So in structural biology, you basically study the atomistic detail of the protein. So this is the nutshell, and I'm working with the eye-related disease and caused by diabetes, that is diabetic retinopathy. So, Ilyas, I would like to know that how you landed up in the field of research, whether it's a sudden parachute jump or it was a smooth landing. So how you ended up in the field of research?
Yeah, I think it's an interesting question because as we've seen before, we all have different ways, I guess, to the same end. And in my case, I guess it followed a bit more. I mean, there is no like big stories, but rather it followed naturally how I got into this field. So I did my studies like you, right? I was in high school and I already had a keen interest in physics. And it followed that. So I come from north of France, right? From there, I went to the undergraduate studies, and I moved a bit down south in Lyon, if you know, the nice ski resort close by. And here I learned about, lots of physics, quantum mechanics, aerodynamics, and statistical physics as well. And it was very interesting there. And I decided to pursue something a bit more specialized. So in statistical physics, I moved to Paris where I did my master's degree for 2 years. And at the end of it, I wanted to continue in this field. And I did, to see the other side, how research was done. And natural extension of statistical physics, which studies, like gases and such, is to try to understand how life emerges at the statistical level. And this field is called active matter. And I did my PhD in active matter. So you can study many things there, like flocks of birds, schools of fishes. And I, from the beginning, started to work with biologists. I studied Neisseria meningitidis, which is a bacteria which can be very harmful because it can invade the brain and give you a meningitis, which is rather deadly. And then I continued on this field to study morphogenesis, so how you start from cells which seem a bit randomly organized and they start to form motion and fully developed organism. And this was in the UK, in Bristol, and then I decided, following one of my supervisor's advice, to apply with my current supervisor. And I came here and then applied for the PASIFIC and study these bacterial colonies, which are, I guess, a natural extension of what I was studying before, but with the addition of growth. And so now here I am, and I'd like to hear about your story, Vineeta.
So I would like to tell you a very small story about my childhood. So how I ended up in research. So I completely remember that day when I was a high grade school student, and principal of my school called my father and told that your daughter is not doing very well in science. And I think she should change her stream.
And then my father came back home and asked me, okay, as your principal told me you are not doing very good in science, would you like to change your stream to some other field, maybe arts, maybe commerce, something else you would like to pursue? And at that age, I just said to my father, no, I would like to be in this field. And at the age of 16 and 17, I think it was a very bold statement which I was making to my father that, no, I would like to be in this stream. And then after high school, it was like I have to opt for medicines. And being a soft-heart person, I was aware that it's not my cup of tea to deal with the humans and this operation, blah, blah. But I would like to be in the field of research. So I pursued the other path. I did my graduation, post-graduation. And to explore more about this field, I did my PhD. And it gives me a platform. It gave me a platform to explore more about research. So I did in my PhD in structural biology, biophysics. But I always have a dream that I would like to work in the field of the translational research. And PASIFIC gave me the chance after coming to Poland that I'm working in the translational eye research. So it's like a dream come true that you always have a dream that, okay, you want to this, and then it comes on your plate. So basically this is how I landed up here, and I'm enjoying the process, and there's much more way to go. So Ilyas, this is my story, but I would like to know more about, like, as you mentioned that you traveled to UK and then now you are here and working with the cell-cell interaction. And like me, an audience, I think would like to know more about what you're working with on your daily basis research, like how your projects meant and deal.
Thank you. So yeah, as I mentioned before, I studied these bacterial colonies, and you can wonder why. So first of all, I would like to have a plate which contains very large colonies to circulate, so that you can have an idea of how big it looks. So there is this is called these cells are on agar, right? So it's a soft gel, and they grew for a bit of time. And this is the object of our study. But even though they look tiny, they are much bigger than what we actually are studying. Because in my case, we start from single cells, right? They are so a single E. coli, which is the bacterium in this petri dish, is 1 micrometer wide and a few micrometer long. And my project is to start from this cell and to track it over its lifetime, and use the computer model to kind of predict where things will go. And this is where the machine learning intervenes, right? So I hope you imagine that this petri dish contains millimeter wide colonies, and even then it contains millions of cells, right? And these cells have, if they don't behave the way they are supposed to, you will start to have problems, right? So let me explain why. In your body, does anyone knows how much cells we have in our body? An approximate figure: 1 million, 1 billion? No. Well, it's much more, right? It's tens of trillions. So it's 1 followed by 13 zeros. This is for the cells which are, well, the cells that you are made of. But there is another types of cell, right? These bacteria actually, they also are contained in your body, and are actually more than those other cells. So the estimates are between tens of trillions to 100 times more, right? There could be 100 times more cells in bacteria in their body than cells. And if they don't behave like they should, then you start to have issue, like if you have an upset stomach, for instance, it might be that these cells, which is called your microbiome, they are not acting like they should, and they should be understood. If you go to the hospital and unfortunately you have to get a urinary catheter. It is known that after a month you're very likely to have an infection, and it's because cells which are foreign to your body start to invade your urinary tract. So this is why we want to understand this. And so with the idea of as a physicist, I'm trying to make a model such that we can study this in a very simple setup. So start from a single cell on this surface, put a glass on it, and we have something that is therefore two-dimensional, and we image it over time. And the machine learning will help us to tell where each cell is, right? And then the idea is to say if the model exactly matches what we obtain under microscope, then the model is a good depiction of reality, right? It's like a very good simulation, and then we can extract number from this model, which are interesting to us, right? These numbers, they are very hard to obtain if you try to poke at the cell with other techniques like atomic force microscopy or something like that. So this is the idea of my project, and I think your involves very different techniques, right? Vineeta, can you tell us more?
Yeah, like as I mentioned that I think we all must admit this thing that eye is the most valuable sense organ. And we all have different color of the eye, size of the eye, but the anatomy of the eye will be same. So I would like to show you some pictures. So how the works deals. So this is the basically anatomy of the eye. I hope everyone can see. So this is the lens part, and this is the retina. So basically, the most of the disease occurs on this part, retina part. So when a person is suffering from diabetes, so over the time with the progression of the age, it starts getting worse. So when the age progress and diabetes is for the longer time, then it seems like your eye will seem like this. So you will have this wool patches on your eye. And over the time when your age will increase, it becomes like this. From like this. So in the diabetic retinopathy, when person is suffering from diabetic retinopathy, what occurs? There is a lack of the oxygen supply and the removal of the carbon dioxide. And to overcome that thing, our bodies start generating new blood vessels, which are leaky and fragile in nature, which start leaking fluid and blood into the eye, and which leads to the loss of the eyesight. So basically, my project deals with this aspect. So when this disease occurs, so basically there are lots of protein involved in the visual cycle. And when disease progresses, there is a decrease of one particular protein, that is retinol binding protein 3. But nobody knows at what level this protein start decreasing. But it gives you an indication that you are progressing towards the diabetic retinopathy. So basically in my projects, I deal with to pinpoint that, okay, from this stage, this protein levels start decreasing, and we want to flag that stage to delay the consequences of this disease. And to know more about this protein, you have to have a structural knowledge of this protein. So basically, we figured out the structure of this protein with the help of the electron microscopy, that is cryo-EM. It's a very, very, very booming technology in the field of the structural biology. And it could give you answer to lots of questions, how to use the protein involved in the disease progressions. So Ilyas, like to wrap up the session, I would like to ask you, as you mentioned that you are working with the artificial intelligence, machine learning, and we all are aware that, okay, nowadays AI is getting a boom and people are using ChatGPT a lot. So I would like to tell you, like, to give a message to our young fellow, what you would like to say?
Yeah, indeed, it's very fashionable now to speak about AI, but I try not to mention it with as a buzzword because in my case it's a tool, right? So like I know Suvik is doing pen and paper, and I use well, most of my job as someone doing physics is involves computation, and the AI part is actually very helpful, but it's just a tool. So I think you should have the idea that it's a tool, and you shouldn't fear that it will become replace you soon enough. I think you still are a generation which will still be able to make a career in hard sciences, because I recently seen talk from a big shot in this field. Sorry, it's French, but Yann LeCun mentioned that these large language models, right, they cannot resonate. According to him, they are still far away from our capabilities of reasoning, right? So they won't steal your job if you go into science soon. So yeah, that's my message. Sorry. What about yours, Vineeta?
So I think I just have one message to our young fellows. Like, I think they have the best example. One is physicist, one is core biologist, dealing with the human problems. And I would like to say that, okay, research gives you a platform where you can explore all your ideas. The path could be different, could be difficult also, because you will feel lots of challenges, but it's really a platform that will give you the wings to fly. So choose your career path wisely and do everything by your heart, by your soul. That's it. Thank you.
Thank you.
Thank you very much. I noted a few keywords when you were talking. So physics, statistics, chemistry, biology, medicine, and also machine learning. It sounds to many like a nightmare. So that must be very complicated. Actually, it came to my mind that if you are ever asked to write an essay about a famous scientist, try Ludwig Boltzmann. It's related to your institute, so it was a very interesting life. You have any questions to our speakers? I'm sure you have. Yes, please.
So I have a question, because you both said something about ChatGPT and AI. Do you think maybe not in 100 years, but later on, does AI will change us? I mean, will replace us? Replace us, or no? Is it not possible?
I'm going to speak as someone who knows a bit about how this works, but also is interested generally in technology, right? So there is this line of thought that either we fuse with the machine, right, or we are replaced. I don't know the time frame. It's very hard to say, but it's possible that we'll have to cohabit with it. I mean, I guess mathematically there is no reason why it shouldn't equate us. So yeah, as a physicist, I would say it's a matter of time, but the time scale I don't know. And regarding the time scale itself, it's very hard to tell, because this field actually is much more ancient than what we know, right? Actually, I think the most influential papers are the basis of machine learning from the 60s or the 70s, right? And it came, it's more and more useful because the hardware is capable of processing a lot of data now. It wasn't the case before, so yeah. This like perceptron, I don't know if you know a bit about it, but these have been conceptualized a while ago.
I'm sure ChatGPT will not write such good essay as you would do. So please.
I just want to comment this situation because I was in this situation. I worked at the international company as the technical writer, and the head of our company one day told that let's fire all technical writers because we have ChatGPT, and ChatGPT can do the work of technical writers. it was 3 months ago. Yesterday, they called me and asked if I could back to work as a technical writer because ChatGPT is just a ChatGPT, and it's not a human being, and it doesn't feel like a human. That's all.
A very important remark, please. You have probably yet another.
Just a comment to this whole ChatGPT thing is like, once we get an information, we tend to accept that information because we trust that information, and trust is a human emotion. So once ChatGPT writes something, we have to come to a fact that we trust that information, and humans are indispensable in that aspect. And so therefore, between a race between ChatGPT and the human thing, I think the human thing will win forever.
Some optimism came. So, okay.
Okay, so I have a question because both of you work as researchers, and I was wondering how every day on this job looks like, and how do you realize different projects that you are given? Like, for example, what do you do? How do you research the data and the reports you give back to your boss and anything like that? Because I'd really like to work as a researcher one day, so I'm very curious about that.
Hi. So it was really a very good question. So I think research comes very organically, and I really appreciate if you want to pursue your career in research. So how it starts, so basically, as I told you that I have an interest in the translational research, so I figured out that area where I want to work. And I saw at my home, my father is suffering from diabetes, so I'm seeing the consequences, and I would like to contribute in this field. So how the things work. So as I said, that this field could be challenging because you have a hypothesis in your head. You will write that hypothesis that you want to do this like in this manner. And sometimes it doesn't work. Then you have to change your strategy. Okay, plan A is not working. Let's not go to plan B. Let's go to plan C. Maybe it will work. And in my case, that plan C also failed. So now I have to go to plan Z, that, okay, let's go somewhere and find the answer. So it will not be an easy path. Sometimes it will be very easy. With the one shot, you will get the results. But it's not always like this. But don't lose hope, because you are not the first person who is working on this field. There are a hell lot of literatures present in the open access where you can get the answer. And like, I think deep knowledge and how you are reading the papers which are already published can give you a hint where you are getting wrong. And then to present to your PI or to present all this stuff to your boss, if you can convince the person, because you are working, it's your hand, you are working. So if you can convince with your work to your boss or to your PI, then you can convince anyone, because I think the more critics you will get, the more beneficial result you will get. So always take the critics in the research work in a very healthy manner. It will help you to grow. It will help you to think more, because your colleagues will ask the most difficult question you never ever imagine. Why it is like this? Why it's not? Why this graph looks like sigmoid? Not linear, and this you never think, but your colleagues will ask, and this will give you a chance to think out of the box. So just explore yourself, and it will come naturally, like, okay, and it will be like it will be a very easy path.
And I have one more question. Would you say that the researcher job is satisfying?
Okay. So I think if you can ask the level of the satisfaction, it varies person to person. Okay, so maybe my satisfaction level is like if I will get the structure and I'm contributing something to the scientific community, it will satisfy me. But for you, I think okay, my mice is not getting diabetic. I'm trying since 6 months and they are not getting diabetic. So it is not satisfying your soul. So satisfaction is a huge term, and so for me, yes, research is my dream and it satisfies my soul. So for me, it is satisfactory, but I think this satisfaction level, satisfaction definition will vary person to person. Sometimes people will not appreciate this, that it is not 9 to 5 job. Sometimes you have to come at 9 and you will finish at 12. So it's not a time-bound job. It's like you have to work. It could be 9 to 2, it could be 9 to 12.
Ilyas, the same question.
If I can answer, maybe from a different perspective, because I'm more a computational person, I would say. I'm not a theoretician, I don't think so. So for me, it's more like a desk job, but with a twist, right? I think it's very varied. I like myself to go to the lab, because I have this microscopy experiment that I need the data from, and I go to lab, and it's very interesting. And it's also, you could see it as an engineering job. I've been working on some side project with my supervisor, which actually is some kind of inspiration to me. So he's a pure statistical physicist by background, but then he moved to more biology subjects, and now he constructs his own apparatus and so on, right? And he likes computation as well, so we're doing many things, and it's true that it's not a 9 to 5 job, but I think it's mostly, you can make it 9 to 5 jobs, I guess, up to some point, but you need the passion, the interest at least. And if I stay overtime, it's because it's really interesting. You have the kind of freedom that you may not have in a regular desk job. So this is the twist. To resonate with Kinga, I think the child asking question, it's kind of a good depiction of what a scientist should be, right? A child with question always popping up. And sometimes you can find, actually, if you look at the state of the art, you might find already answers to something you encounter. You do an experiment that something goes wrong, actually maybe it has been reported, right? And this is where there are papers, right? So you Google or you go to the archive or some specialized journal and you have a look and you learn something. So yeah, that would be my take on it.
Thank you very much.
In short, what they said, it's not a job, it's a hobby that they pay you for. So that's a very dream world, I would say. Please.
Hi, I'd like to ask the question to both of you and honestly to all the scientists in the room. I'm very curious how it works, like how to live of the job, because a scientist is not a job like a 9 to 5. You can't just copy files because you invent something new. So I wonder if the main source of income is research funds, or do you have your employers that pay you for researching on a certain topic? I'm really curious how that works.
So it works on, at our stage, since we are still postdoc, I think most of us, it works by getting grants, right? Either you get a grant like we did, so this PASIFIC grant, we wrote it and we got selected, or it's your supervisor, right? We got a grant and then you're hired, but it's a temporary contract, right? And then later on you can get a position where you'll be, you will have a salary, but you still need to get grants to fund your research. Right? So it depends. If you're pure pen and paper, I think you can live only with your salary. Some people do it like that, right? But if you're running a lab like in my group, you need a lot of money, right? Like I think millions of euros. And so this is why you need to write competitive grants. And this can be tedious because it takes time and you may get rejected, and it's very stressful, I guess, if you don't have the money. So that's one of the drawbacks, I guess, but this is how it works nowadays.
So same answer from my side, you have to write the grants, and then after winning that grant, you will have your own salary. You will have, so how the research grants are bifurcated. You have one salary, like your own salary or the team you are taking into the account that, and then consumables which you are going to consume in your research projects, and then some other costs. So there are lots of costs. It's not like when you get a grant, it will comes only with the salary. It comes with the other money also. So when you see, when you will start applying, you will see that, okay, this grant costs 1 million. So it's not like that 1 million will come to your pocket. It will comes with its own cost bifurcation that you will have. Apart from your costs, it will have lots of other salary. And sometimes your PI has the project, and he and she can hire you as a researcher, as a technician, or as a PhD postdoc. So basically, and using that money comes within a responsibility, because it's a taxpayer money what we are using for the research. So it comes with its own responsibilities.
I want to make some comment and maybe answer for for you guys. I think there is a wrong idea that there is a, the very, very different aspect in the academia. Like we are postdoc, but there are professor, there are assistant professor, there are associate professors. So these are all different terms. Now what we are having is just a temporary position for 2 years, but it doesn't mean that you are not going to have a permanent position in a university or in an institute. So when you are having a permanent position, of course, you are having a regular salary, sometimes which is really good, and at the same time, you can support this salary with the grants, which is like writing and the proposal. What we want to say by the proposal, actually, it depends on your research area. For example, I'm a migration scholar, and I wrote a proposal related to immigrant entrepreneurship, and I got this grant. And if I am having a, the permanent position, again, I can apply for another grant, and I can support my salary with the grant which I am taking to. So don't be that much hopeless that we are just having a temporary position and it will never turn to the, permanent one. It is just taking time, but definitely it is going to happen one day. And that's why if you are, thinking that, and at the same time, when you are doing a science, I think you have to be, ready for the challenge. We are not doing it every time for the money, but we are doing it for the betterment of the humanity and for the, betterment of the world. So I hope it can give a clear answer actually what we are doing and what we are trying to do. Thank you.
Thank you very much. It's a bit, we can compare it with like sport, yes, that we always compete. But if you are good enough, then you are not poor. So.
To add something, yeah. I think you will have time to. Don't be worried about this grant stuff. If you get into science, you'll have time to get into the process, right? It's a lengthy process. Don't worry.
I just wanted to apologize. I didn't mean to come off as saying that you only care about money. I was just mostly curious, how as an artistic job, because you invent something as a scientist. And I know that artists have it hard, so I was just curious. I apologize if I came off offending someone.
It's okay, don't worry. No. It was a really nice question, and to be very honest, being worried about your financial position is not at all a wrong thing. You could do the work if you are financially satisfied. So don't worry. Don't even feel sorry for it. So don't worry.
I have a question about the environment in the job, because I imagine that being a researcher and sitting at the desk all day can be quite isolating. I wanted to ask if you work in groups of a couple people, or if you work by yourself.
Okay, so in my case, we are the group of 5 people, and and we all from different nationality also. So sometimes you work within a group, and as I'm working with my project, so I'm working on it alone. But it more or less, it's a team job. When you write a project, or when you are in a project, you cannot do each and everything by yourself. Okay, I will do this task, everything I will do by myself. It's not at all possible. You need help, you need a team, and you need a support system. So definitely sometimes you have to work alone also, and sometimes it becomes very tiring. But you know that your team is there, your support system is there. So it's like research is basically not an individual effort, it's a team efforts in one direction. So I think it's more about team rather than in isolating and feeling isolated. It's not like this.
So my experience is, I guess, a bit different. I was in very small teams. During my PhD, I was mostly alone, and yeah, it can be a bit isolating. But I guess it depends of your mindset or your personality, right? Some people actually like the freedom. So some supervisor will force you to come, you know, even if you don't want. And reciprocally, some PhD students, they'll be very good alone, right? They will work from home, never come to lab, and have tons of papers. So it depends. But no, I'm in a big group and I quite enjoy it. I could work from home remotely most of the time, but I enjoy the social experience, you know. So and actually collaborating as well. Yeah. So yeah, it depends on how you feel with being around people.
Actually, today is part of their job as well. It's a very teamwork. You're a big team, so.
Thank you very much.